Okay, so now they know what the organism is. Is that good enough? Well, that's a start, right? It makes some sense. But wouldn't you like to be able to grow the organism, to do something with it, to work with it with your hands? These days, you would probably next just sequence this whole genome, right? Take a sample, wash it as much as you can, sequence it, get millions of sequence reads out of it, assemble the entire genome of the organism. You've got a ribosomal sequence so you know which genome you get is the correct one. And then interpret the genome sequence to tell you what kind of a living it might make and all that kind of stuff. That was beyond their capability at this time. And so they wanted to grow it. Now this is not the same people. This is Carl Stetter's lab. Carl Stetter ran, he doesn't anymore. Probably the single best lab in the world at the University of Regensburg for isolating weird organisms, weird microbes. And this is Robert Huber, who now actually runs that lab, and some of his other folks. This is called a monogram. So this is a paper that formally describes the organism, gives it a name, Thermocrinus ruber, and registers it as a type strain, as a definitive identification of an organism. Monograms have been used for a very long time. You can look in old museums and things, and every new species, there's a monogram that describes that species. This is how they do it in microbiology. Thermocrinus ruber, species novel, genus novel, so it's a new genus and a new species, a pink filament-forming hyperthermophilic bacterium isolated from Yellowstone National Park. So they were successful in growing this organism. How did they do that? Nobody else could do it before. How did they know how to grow it? So the previous folks had all based their isolations, the enrichment media and cultivation media, based on the physical properties of octopus spring, right? So deep chemical analysis of what things are in this spring, and you try to replicate that. And they fail. Is there something you know now that you didn't know before that might help you? Let's go back to the prior paper. Here it is. Is there something about aquifex and relatives that might help you grow it? Every aquifex, every member of this group, at the time and still today, are hydrogen oxidizers. Every single one. They carry out the null gas reaction. And so, if you're going to grow an organism in this phylogenetic group, you've got to give them oxygen, you've got to give them hydrogen. Not the sort of thing you see in a chemical analysis of water, because those things evaporate, and they're at low concentrations to start with. And so, these guys did enrichments based on those conditions. They actually did some, they don't describe it very well in the paper, but they did what I think is really cool. It's kind of like a little microarray of enrichment culture. So they start by trying to do an enrichment. And so they've got these bottles, these pressurized bottles of media, and the media is based on the chemical composition required. They're present in Yellowstone Park, and then also they add some supplements just in case. And they then incubate. They did hundreds of these bottles, and they had them in a big cube with one variable in each of 3 dimensions. Temperature, so from 75 to 90, I think, and then varying the oxygen concentration and the hydrogen concentration. So you have like a 3-dimensional array of possible growth conditions. And they inoculated them with pink filaments, yeah.
Did they vary other environmental conditions at all, or only the ones that they had?
Oh, they tried everything,
Okay.
but these are the ones that worked. Now, some of their bottles turn pink. That's a good sign, right? But when they looked at them, this is what they saw. Rod-shaped organisms. Okay.
So they were inoculating the sample
From Yellowstone. Yeah. So how do you know what's going on in these enrichment cultures? You use fluorescent in situ hybridization. So they took samples from these things, and sure enough, these rod-shaped bacteria would light up with their probe. So they're pretty sure they've got the organism, even though the phenotype isn't right. Well, how are you going to isolate this thing in pure culture? All you have now is a bottle that's got your organism in it, but it certainly has lots and lots of other organisms as well. Well, they used, and this is where they developed it, the same trick that they used for nanobacillus later, and these are optical tweezers. So they know what the morphology of this EM7, or the thing that lights up with the EM17 probe is in these enrichment cultures. And so what you do is you've got a microscope that's set up with an infrared laser. And the laser shines down through the sample, and the sample is in a capillary tube. And so you take some of your enrichment culture, draw it up in the capillary tube, and you find a cell that looks like the ones you think are right. Now when you do in situ hybridization, the cells are dead. So you can't label them in advance and pick ones that are labeled. So you have to do some that look right, you do several of them, and hope that one of them really is the right thing. And so you find a cell that looks right, you turn on the laser, or swing the mirror into place, and that laser will grab it and will hold that cell in place. The physics behind that, by the way, is not very well understood. You get arguments about why optical tweezers work. But then you take some sterile buffer or media, and you gently push that through the capillary, and everything else blows away. Your cell, though, is still trapped in these tweezers. Then you can turn the laser off, draw the syringe back, and now you've got one cell in your syringe. You inject that into media grown of the same kind that your enrichment culture was done in, same oxygen concentration, same hydrogen concentration, same temperature, and it grows. Now, it wouldn't have to grow, right? You might imagine that it needs some nutrients, vitamins or something that the cells, other cells in the community are providing it. But in this case, it grows. So you get lucky. But again, what you get is this thing. Rod-shaped cells, and more specifically, flagellated cells that at the appropriate temperature will swim around. They look nothing like a filament. And so you don't know what's going on. Is this really the right organism? It lights up with the probe. So fish out its ribosomal RNA and sequence it, right? And make sure that it's the right thing and not something that just happens to hybridize to your probe. Well, when you do that, oh, here's some more EMs. It just shows the cell structure. It's got an outer membrane, cell wall, inner membrane, etc. Periplasmic space, yes. It grows very nicely. So this is doubling time. And so the doubling time is lowest, that is, growth is fastest at 80 degrees. It won't grow above 85. And it doesn't like anything below 65 or so. When you fish out that ribosomal RNA, what you find is that this organism is really, really close to EM17, but it is not identical. This is to be expected. It turns out that if you look at a single species in a microbial community, you end up having a diversity of related organisms. It would be really unlikely for you to get exactly the same creature when you pull it out this way. It's like they pulled out an eastern or a gray squirrel instead of a red squirrel. Nevertheless, it's pretty close. They've got the right thing. How then do you explain this filamentous growth in nature and the rod-shaped motility in culture? Well, their thought was maybe it has to do with the physical conditions of the spring, not the chemical conditions of the spring. You all, I'm sure, have seen pictures of plants that look very different, grown in different environments, right? And so they built a new octopus spring, or at least a reasonable facsimile. And so they built a glass box, and they would pump media in on this side, and it would fill up with media, and there's this ramp, and the media would flow over this lip and down in here to an outflow. And so they would start by putting some of this EM17, or they call it Thermocrinus ruber. They inoculated it here, they start to pump media in it, and sure enough, right here at this turbulent overflow, the organism would fasten to the glass and grow its filaments. And it looks exactly like the stuff in octopus spring. And so they do have the right organism. This pleomorphism, this ability to look different under different conditions, is quite common in the microbial world. We think of organisms looking all the same. E. coli is a short rod, right? But organisms in nature are a lot more diverse than this. There's another story of this spore-forming filament in insect guts. It's quite common. I mean, a wide variety of insects have this filament, this organism in their gut. It's called arthromidas. And if you try to grow arthromidas on plates, it doesn't grow as filaments. It grows as individual rods. It is, in fact, Bacillus. Bacillus is filamentous in its natural habitat, the insect gut. But in soil and in culture, it grows as individual rods. And the same thing is going on here. And so the paper goes on to, like any monogram, to talk about things like metabolism, how does it make a living, effects of light. And so you need light in order for that pink color to come out. It's probably an accessory pigment that's used for protecting the organism from UV light. A whole series of things, DNA-based composition. Why on earth would anyone think to do a DNA-based? Well, who cares what percent GC the organism is? It's genome. This is one of the standard microbial characterizations. And so in order for this to be formally described, that's just a thing you have to do. It's antiquated, but that's what it is. And yeah, so in other words, so 47.8 mole% GC. So DNA contains GC, A, and T. And so there are 4 numbers. There's going to be some fraction of each, and that should add up to 100. Maybe it's 25% each, right? It never is. But for every A, there's a T. And for every G, there's a C. And so the GC, the fraction of G and C will always be identical, and the fraction of A and T will always be identical. And the 2 will always add up to 100%. So one number is sufficient to describe the base composition of DNA. And so it's 47.8% by mole, meaning just the numbers, not weight or anything else. And so it's 47% GC, which means 47.8, which means that it's 52.2% AT. And that means that it's, whatever half of that is G and whatever half of that is C in the DNA. That's all that means. And so they named this organism Thermocrinus ruber. Hot hair, red, in Latin. I'm going to finish with one little thing. We'll go back to this paper. How many of you guys have isolated organisms in a research lab? Anybody? You? Okay. So you're working with colonies on a plate, and you have to call them something, right? You don't know what they are, you have to call them something. And so in this paper, Anna and Jean called these EM, and they just number them. EM1, EM2, EM3, EM4. Each one was a colony on a plate, which was an E. coli clone of a ribosomal RNA. Sometimes these are pets' names, Rover 1, Rover 2, whatever. It can be anything. People get original. Maybe they're like car names, right? They can be numbers and letters, or they can be names. They can be whatever people want. EM17. Why EM? The clue to this is in the acknowledgement section. So here's the acknowledgments down here at the bottom. We thank such and such people for this. We thank other folks for money. Oops. Special thanks to Douglas Adams. How many of you guys know who Douglas Adams is? Oh, come on. Hitchhiker's Guide to the Galaxy, right? And his Electric Monk, EM, Electric Monk. They happen to have this book with them when they went on this expedition. And one of the characters saw everything in shades of pink at one point. And it's completely ridiculous, right? It doesn't mean anything. But when you're on an expedition in Yellowstone Park for some weeks, that's what happens. If you ever have a chance to read Dirk Gently's Holistic Detective Agency, it's his best book, best work. And that's where the Electric Monk comes from. That's it. Wednesday we'll be talking about, I don't remember what we'll be talking about. Oh, the scaly snail. There's a snail that lives down in deep sea hydrothermal vents in various places, predominantly the Indian Ocean, that doesn't have an operculum, doesn't digest food, doesn't eat. It's an autotrophic animal, and we'll talk about that next time.